Right, it's 11 o'clock. Let's get going. Today we are going to cover, a lot of material, and this is the first of two big topics in lecture exam 1, and this is on metabolism. Again, this is a lot of biochemistry. Your textbook has this material at the level of a biochemistry class. I want you to concentrate on the summary figures, not the details of the biochemicals. That's going to be more summary type things that I'm going to be talking about. Now, I mentioned some of this before, and I want you to pay attention to this, because this is how you're going to do not just this topic, but any topic in the future that looks intimidating. You're going to resist the temptation to be intimidated, and you're going to do these two steps. You're going to take a step back and see the big picture. Things like, what is this for? Why are we learning this? What is this whole thing about? What is this for? Well, in this case, this is about how our cells turn nutrient links into molecules of ATP, because that's the only type of energy your cell directly uses. When we say that glucose is the most primary form of energy used by the cell to make ATP. ATP is the only thing that the cell directly uses in its cell processes. And then break it up into small sequential steps. The whole thing is intimidating. Don't learn the whole thing. Start at the beginning and learn the sequence of steps. Each bite-sized step you can learn. Just know that you want to keep it in order, because when you are done with that, boom, you have it, the whole thing, and you weren't intimidated. So we're going to concentrate now on 3 sequential steps, starting with this particular nutrient link. Now let's go back over here. There are 4 biologically important macromolecules. I call them chains. Well, out of the 4, only the first 3 are nutrients. That's all we're talking about. And that makes sense, because actually ATP is a link of RNA, a nucleic acid. Okay? Now, remember, there's catabolism. When you break down large things into small things, you release energy. If you have to build things up, you got to put energy in, and that's anabolism. But I pointed out, too, that this catabolism from chains to links is what your digestive tract does when you eat that cheeseburger. Once you have the links, and that's what's absorbed into your blood, this is the 3 links, how our cells turn nutrient links into ATP energies. I'm just going to start with glucose, and it's not making ATP from scratch. It's just putting the last phosphate group on ATP. That's how it's making ATP. I'm going to start here, and then after I have that framework, then I'm going to add the other 2, and boom, we're going to cover this in 1 lecture. It's doable if you take these steps. Don't just do this for metabolism. Do this for when we talk about protein synthesis. Do this when we're talking about any other thing that looks really intimidating. Okay? Any questions before we get started? I cannot share the recording with the rest of the class. That turns this class into an online class, and then it doesn't qualify for your prerequisite. Okay, let's go here. So we talked about the 3 nutrients last lecture. I'm gonna have to believe you guys got it. Otherwise, there's no way we can get through it. We gotta know about the 3 nutrients molecules: carbohydrates, lipids, and proteins. Okay, the lecture exam 1 isn't for a couple weeks, sorry. So, but this is going to be a really big topic, and actually the last lecture was included in this because it's talking about the nutrient molecules, as well as things like catabolism and anabolism, things like condensation reactions versus hydrolysis reactions. That's all part of this. And also we talked about this too. I don't want to get too far away because this will be relevant pretty quickly. Remember that we have to talk about how a lot of energy to make this ATP is going to be in the form of high energy electrons. And this is where we have to use these dinucleotides. And these are actually also RNA nucleotides, but there's two nucleotides, and that's why it's called a dinucleotide. And remember that the high energy electrons are not free electrons. They're in the form of hydrogen, because all hydrogen is an electron and a proton. So if you see the H's, it's got the high energy electron on it. If you don't see the H's, that's the low energy or oxidized state. Remember, we talked about redox reactions. And I said, well, if you gain the high energy electrons, you're reduced. And so that doesn't make sense unless you remember that electrons are negative. So if you're gaining a negative, you're reduced. Okay? That you got to have down as well. Otherwise, none of this makes sense. And in the lecture packets, you want to get that open to page 9. And you may also want to get out the document for the potential lecture exam 1 essays, even though they're going to be in fill in the blanks. We're going to basically finish that today. I'm going to cover the rest of them today. All right, now let's get going. All right, remember, here are the 3 sequential steps. We had already talked about the 3 sources of glucose, since we're starting with glucose. And the 3 sequential steps are glycolysis. The Krebs cycle, there is one reaction just prior to the Krebs cycle. We'll call that the intermediate step. Some textbooks call it the linking step. Some call it the Krebs cycle prep step, but there's one reaction just prior to the Krebs cycle that's in between 1 and 2. But the vast majority of the ATPs is actually coming from the 3rd step, and this is the electron transport system. Now, we talked about also the term phosphorylation, putting that last phosphate group on ADP is called phosphorylation. And there are 2 forms. Substrate level phosphorylation, and I told you that the substrate are the carbons. So if you have carbons, that's substrate level phosphorylation. This was covered last lecture. If you make ATP where there are no carbons, did my camera go out?
Yeah, your camera went off for some reason.
Oh, there it is, okay. But if there are no carbons there, that is, there we go.
There we go.
Oxidative phosphorylation. And that's happening in that 3rd step. There's that term, oxidative phosphorylation. Because there's no carbons here, you're going to see pretty quickly all the carbons, all 6 carbons from glucose are burned up into CO2s in steps 1 and 2. No carbons left in the 3rd step. That's why that's oxidative phosphorylation. Okay? All right, where did I put my... Okay. Okay, any questions before we start? All right, we covered all the preliminaries. I just reviewed them. Now let's do the 3 steps, starting with glycolysis. It literally means glucose splitting. It is the first of the 3 sequential steps. You do have to know that this is happening in the cytoplasm. So here is glycolysis on the whiteboard. I'm just going to write step 1. And this is happening in the cytoplasm. Okay. So you get a 6-carbon glucose. I'm just going to abbreviate it on the whiteboard as 6C. And then you got to put in 2 ATPs in, and this is in the animation, because you have to phosphorylate glucose with high energy. Otherwise, nothing happens in the cytoplasm. Once that happens, then a whole sequence is going to happen, and you eventually get 2 3-carbon molecules. Now, you're going to see this written 2 ways, and I want you guys not to worry about it. And it has to do with this carboxyl group that I talked about last lecture. The C double bonded oxygen and then the oxygen, and then a hydrogen. If there was, it's written like this without the hydrogen, it's called pyruvate. It's pyruvate ion. If you put the hydrogen on it, it's pyruvic acid. Now, I don't want you guys to be, caught up in that. Don't get your underwear in a knot between pyruvate and pyruvic acid. For our purposes, they're equal. Just one has the H on the carboxyl group, this one, the other one doesn't. Same thing here, I wrote oxaloacetic acid. You'll also see that as oxaloacetate. Same thing, it's just this one has the hydrogen on it. Everybody got it? But that is not crucial, so we will just call them equivalent for our purposes here. So that's the glucose splitting. 6 carbon goes to 2 3 carbons. Everybody see how it's glucose splitting? Glycolysis. There's a couple other things that you need to pay attention to that's happening here. Is that number one, you don't need oxygen. It could be with or without oxygen. Okay? With or without oxygen. And that means it could either be aerobic with oxygen or without oxygen, anaerobic. But the other two things that are going on here is that you got to put those 2 ATPs in, otherwise nothing happens, and you get 4 out. But because you had to put 2 in, we will just count those as 2 net ATPs. 2 in, 4 out, you have 2 net ATPs from glycolysis per glucose. Okay? Also, you put in 2 NADHs, and you got, 2 NADs, and you got 2 NADHs out of it. That's in purple. Everything else, you really don't have to know all the specifics. So like, look at this box in the middle of the slide. It's showing you the whole thing, starting with the glucose. Here's the glucose phosphorylation. The 2 ATPs are going to go in, and then it changes it. Now it becomes fructose. Here comes the second ATP in. Now it has 2 high energy phosphate groups, and now it can split. But it's going to not go straight to pyruvate. It's going to go through all these intermediate steps that you don't have to know about it because this is not a biochemistry class. You just got to know the main things in and the main things out. Okay, here's 2 of the ATPs coming out. Here's the other 2 ATPs coming out. So 4 out, 2 in, 4 out. That's 2 net. And you're done with 2 pyruvates. Okay?
Question?
Yes.
So for the 2 NAD to the 2 NADHs, is there just a standalone hydrogen that's a net from like after?
Yes.
Okay.
Yes, and it's not connected, and that's why I write it out as NADH plus H, but there's a second one. Now here's the thing, people. Again, this is not a biochemistry class. On the lecture exam 1 on Canvas, can you recognize the high energy and the low energy? You could see some people will just write this out as NADH2. It actually isn't, don't matter for our purposes. Everybody got it? And actually, NAD is actually an ion, NAD plus. Okay, I don't care if the plus is on there or not. Everybody got that? Now, if you look on the potential lecture exam 1 essays, this is lecture exam essay number 2. Glycolysis. Where does it occur in the cell? Cytoplasm. What goes in? There are things that go in. What comes out? There are things that come out. No intermediates. Everybody see that? And this is substrate level phosphorylation because these 2 net ATPs, look on the whiteboard, these 2 net ATPs are being phosphorylated where there are carbons. There are carbons here, substrate level phosphorylation. Carbons are the substrate. Boom. That's glycolysis. This is what your textbook has. Biochemistry. Hey, I had to take biochemistry. I had to reproduce this from memory, including the formulas. You don't have to do that. You have to do this, the summary. Everybody got it? Any questions? Okay, now because this one can be done with or without oxygen, I'm going to have to branch out right here first, just for a second, and talk about anaerobic respiration. And it's really easy because it's only one step reaction. And what I'm going to do here is I'm going to put this artificial division because I don't want to have to write this twice on the whiteboard. But this is going to be with oxygen. And this is going to be without oxygen. But in reality, both halves will be doing the same thing. If you have oxygen, both pyruvates are going to be doing this. If you don't have oxygen, both pyruvates are going to be doing this. But do you see why what I'm saying, so I don't have to write this completely on the whiteboard twice? I'm splitting it to save my hand and time. But it's either going to do, it's either or, and both will do it. There'll never be a situation where one will do this and one will do that. Does everybody understand the words that are coming out of my mouth? Stay engaged because I'm using 7th grade level English. Everybody understand the words that are coming out of my mouth? I'm writing it once and splitting it. That never happens. They're either both going to do this or they're both going to do that. But man, I only have so much whiteboard and only so much time. All right, so let's talk about what happens when you don't have oxygen. Your cytoplasm has an enzyme called LAD, not Los Angeles Dodgers, but lactic acid dehydrogenase. And because of that, without oxygen, boom, it's going to just make another 3-carbon lactic acid. Or if you take off the little last H, it's lactate. Means the same thing. The bummer here is that to do that, it needs energy. And what it's going to do is it's going to use the 2 NADH plus H that it got from glycolysis and turn it back into the low energy NAD. One reaction, and it is because of this enzyme lactic acid dehydrogenase. Boom, one reaction, it's done. Now, if we had the same enzyme as yeast cells have, we wouldn't make lactic acid, lactate, we'd make ethanol, which is why anaerobic respiration is called fermentation. Those are synonyms. Wouldn't that be great if we had that enzyme? We'd all be cheap dates. You guys want to have a party? Let's all run around the track twice. We'll be all shit-faced drunk. But no, we have lactic acid dehydrogenase, and instead we feel the burn. Everybody gets the difference?
Yeah. Isn't there something called brewery syndrome where the gut bacteria like brew all the carbs in your body into alcohol? I read something like that.
Yeah, your audio is breaking up, but yes, the gut bacteria can do other things. They have a different metabolism, especially as prokaryotes. And that's why when they take the food, and they're using the same food you are when you swallow, they'll do a bunch of different things, including making methane. So you don't do it, that's your gut bacteria. All right, so for this step, you will only get 2 lactates. Again, that's the same as lactic acid. And the 2 net ATPs from up here. The way to look at this on the whiteboard is that if both sides are doing anaerobic respiration, okay, these 2, I gotta go this way. And that's because one is on one side and one is on the other. This purple is going this way, and this is going back here. Doing a circle, but I got all that over there, so you're just gonna have to connect it yourself. Boom, boom, which means the only thing you get for that glucose is 2 net ATPs. This high energy electrons, that's what's supposed to go to the 3rd stage, that is over there on the far right. Well, you can't even do that because you burn it up making lactate. So you hardly get any ATPs with anaerobic respiration. Just the 2 from glycolysis. It's not very productive. Okay? Because of this, most cells will only do this in an emergency. The only cells that do this routinely is skeletal muscle, we're going to talk about in a second, and red blood cells, because when red blood cells are in venous blood, they have low oxygen. They're used to doing this. But no worries, they're going to go through the right side of the heart, go to the lungs, and get oxygen. And besides that, RBCs have a very low metabolism anyway. They're hardly doing anything. Just carrying oxygen. Any questions on anaerobic respiration? Here's that loop. Here's that loop. NADHs that come from glycolysis, nope, it's getting burned to make lactate or lactic acid. That becomes NADH again, and then nope, boom. And so for each glucose, you're only getting 2 net ATPs. All right?
You said it would only be used in an emergency by the body. What kind of emergency would constitute the body switching over to only anaerobic respiration?
Getting stuck underwater.
Oh, okay. If you can't breathe, it starts doing that.
Right, exactly. If you have, get something stuck on your cricoid cartilage in your larynx, and no one knows how to generate an appropriate sense of urgency and do the Heimlich, your cells are going to start doing this as an emergency.
So the body switches over pretty fast then from one to the other.
Yes, it can. It can switch over very, very quickly. But again, most cells normally won't be doing this. Skeletal muscle cells, especially if you're out of shape, you can get up out of your chair and go to the bathroom. Some of your muscles are already doing anaerobic respiration if you're a couch potato. Okay, so this is talking about RBCs have low metabolic needs anyway. They even lack mitochondria. And skeletal muscle, we're going to talk about that when we get to muscle physiology, but they're a freak, okay? If you're looking at your regular body cells, if they double the consumption of ATP energy, the rate of consumption, that cell is working really hard. Well, skeletal muscle cells are just laughing at that, because when they go from rest to working, skeletal muscle can increase their demand 100-fold. And if you're in good shape, over 100-fold. So their skeletal muscle is just in a league by itself, and that's why they will do anaerobic respiration routinely. And that's why we got to study the Cori cycle, which is on your essays, essay number 3. And if you look at the words of this diamond and look at the words of that number 3, it's exactly the same, because I don't want to be confusing, I don't want to be straightforward. What is the Cori cycle? Because you're only getting 2 ATP per glucose, what your body wants to do is it wants to recycle glucose. So here's the muscle doing anaerobic respiration, getting the 2 ATP. The one step that's happening in the muscle cell is going from pyruvate to lactic acid. That's why you feel the burn. Lactic acid goes into the bloodstream. Your pH actually starts to drop, as well as from not just the lactate, but from the increased carbon dioxide. While your liver catches the 3-carbon lactate, puts it back into pyruvate. Pyruvate is not a carbohydrate. Your liver will actually put energy into this and recombine it into the 6-carbon glucose. When you make glucose, new glucose from non-carbohydrate sources, what do you call that? It's written twice on the slide. Once in the figure, once in the paragraph.
Gluconeogenesis?
That's what it is, new glucose from non-carbohydrate sources. So people, this is what I want you to start doing, is I want you to start studying after every lecture, because this was actually covered from the last lecture. 3 sources of glucose: facilitated diffusion. What's the second one? You're splitting off glucose links off of glycogen.
Glycogenesis?
Good try. It's glycogenolysis, splitting apart glucose. Lysis is breaking apart, so you're splitting apart individual links off this huge branch chain we call glycogen. Glycogenolysis. The third is this one, gluconeogenesis, making new glucose from non-carbohydrate sources. All right, so what it does is it releases free glucose into the blood. That's what's traveling through your blood. It was actually phosphorylated glucose from this energy, but it takes off that last phosphate group and spits out free glucose. It goes into the muscle where it has to be phosphorylated again to do glycolysis. And it does it again and again and again, and you're able to do anaerobic respiration longer because you're recycling the glucose. Getting only 2 ATP per glucose, you're going to expend your nutrient energy in a hurry. Here, you can make it last a little longer by the Cori cycle. Okay? Any questions? Feel the burn. What does that mean? Probably the hardest event in track and field is the 800, because that is sprinting for 2 full laps around the track. Even world-class athletes, their bodies are on fire from acidity, lactic acid and CO2. And that's why even though these are world-class athletes, when they cross the finish line, they're kneeled over, maybe on their knees, and they're breathing as fast and as hard as they can. Okay? Well, if they didn't have the Cori cycle, they would have never lasted 2 laps. There, everybody got what this is for? You can only do this for so long, anaerobic respiration, but with the Cori cycle, you can do it longer. Any questions on anaerobic respiration or essay number 3? And you're not going to reproduce or replace the glycogen you burned up with glycogenolysis unless you eat something. And that's still going to take a few days. Okay, that's the first 3 steps, and it's the only step that happens in the cytoplasm. The rest of the steps are going to happen in the mitochondria. And because to do the rest of the steps, you got to have oxygen, we're going to now finish up aerobic cellular respiration. You must have oxygen or you don't do steps 2 or step 3. You only do step 1 and this one reaction when you don't have oxygen. That's it. Second of 3 steps is called the Krebs cycle. I'm not going to make you learn the names of all the intermediate steps. They're as a group called keto acids, but you do have to know this 4-carbon molecule I'm going to talk about in a second. This thing that actually enters the Krebs cycle, and the first product. So basically the first reaction of the Krebs cycle, you do have to know all the substrates as well as the product. And that's because some textbooks don't call it the Krebs cycle, they call it the citric acid cycle. And I need to at least give you that, so if you ever see that, you're not completely lost. But it's the second step where all 6 carbons have been now broken down into individual carbons of CO2. One 6-carbon molecule into 6 CO2s. This is the final catabolism that is occurring in the cells. And then the last step is the electron transport system. And that's also in the mitochondria. So let's go ahead and review what's on this slide as far as the parts of the mitochondria, because you do have to know mitochondrial anatomy for these words to make sense. Remember that a mitochondria has 2 membranes. This is the mitochondria, has 2 membranes. It has an outer membrane and an inner membrane. Well, in the center, inside the inner membrane, is called the mitochondrial matrix or matrix. But the space in between the inner and outer membranes is called the intermembrane space. You've got to know this to understand the third step. In fact, if you look on page 9 of your lecture packet, that also requires you to know this anatomy. Way up on the top left is the cell membrane and cytoplasm. Then you're in the mitochondria, and you can see it has the outer and inner membrane. The very center of the mitochondria is called a matrix, and the space in between outer and inner membrane is called the intermembrane space. What it shows you there too is you will have these little fingers of inner membrane because it's so hyper folded. So each one of those fingers is called a crista. So I'll do this, so hopefully it won't be. It's just a finger or fold of the inner membrane. And that's what you see on page 9 of your lecture packet. Okay, does that help? As we're talking about inner matrix, and the electron transport system is on the inner mitochondrial membrane. We are done with step 1. We're ready to go to step 2. Are there any questions?
Does the mitochondria have its own DNA?
Yes. Its own DNA, and they reproduce like bacteria when your cells are faced with chronic acidity. You feel the burn, lactic acid, from this enough, your mitochondria will actually grow. And when they get to a certain size, the DNA will replicate, and it's in a ring just like bacteria. And then it'll split and divide and become 2 mitochondria. As long as you keep feeling the burn, those 2 go to 4, those 4 go to 8, and they each just get bigger and bigger and bigger, and those cells now can make a lot more ATP. We'll talk about
of
that.
This entire cycle, the electron transport is the one that makes, what, 32 out of the 36 total?
Yes, that's where the big payoff is. But we'll get there. Any other questions? Okay, I told you that the Krebs cycle between steps 1 and 2 has an intermediate or linking step, so we got to cover that first. And we actually have seen this already in week 2 PowerPoint. I just didn't call it that. Okay, so this pyruvate, the 3-carbon pyruvate, goes all the way into the center of the mitochondria, crossing both membranes. There is the first decarboxylation reaction. We covered that on Wednesday. Decarboxylation reaction, and it loses the CO2. The 3-carbon Now becomes a 2-carbon. And the 2-carbon in organic chemistry is the acetyl group. Well, the acetyl group gets attached to a coenzyme A, and it becomes acetyl-CoA. And this figure I used last week to show you decarboxylation. 3 carbon goes to 2. The 3rd carbon came off as CO2. Well, that is catabolism. You've gone from a 3 carbon to a 2 carbon plus a 1. That's catabolism. Now there's energy available because it's released and it goes into high energy electrons on NAD. And you get NADH plus H. That coenzyme A is going on it and it becomes acetyl-CoA. This is what I have on the whiteboard right here. This part right here is the linking step. 3 carbon, 1 CO2 comes off, becomes an acetyl group. You're going to get high energy electrons from that catabolism on the NADH plus H, and then coenzyme A comes in and it becomes acetyl-CoA. And acetyl-CoA is what enters the Krebs cycle. Okay, and that's occurring in the mitochondrial matrix. Okay, acetyl-CoA is what enters the Krebs cycle. So this is the one reaction step between step 1 and 2. And they didn't want to call it its own step, so they just call it the linking step into the Krebs cycle. Again, maybe you should write this next to this to tie it to the previous slide. This only happens if you have oxygen. If you don't have oxygen, you're not even going to do the linking step. And on the whiteboard, if you don't have oxygen, you're not going to do that. You're going to do this. Boom. Okay, linking step. Now let's talk about the Krebs cycle. This again is happening in the center of the mitochondria, in the mitochondrial matrix. That is coenzyme A breaks off and it just drops off the 2 carbon acetyl group onto the 4 carbon oxaloacetic acid or oxaloacetate. Same, means the same thing. I will have you learn the first reaction. 4C plus 2C equals 6C. And that 6 carbon molecule is citric acid. I need for you guys to learn at least that. I'm not going to make you learn any of the isocitrate and alpha ketoglutarate or succinyl-CoA. I'm not going to make you learn the rest of them. Just that first one, because some textbooks call this the citric acid cycle, and you need to know why. It is the first product of the first reaction. Again, you don't need to know all this. You don't even need to know the order. When I took biochemistry, we had to not only did we have to know all the chemical structures and names of all the intermediates, we had to know the names of each enzyme in the cycle. So I know what I'm teaching you is hard, but it's not that hard compared to what it could be. Okay, just telling you. All right, for each cycle, you're going to get 1 substrate level phosphorylation that happens down in here. Put it in green. You're going to get 3 NADHs, 1, 2, 3, and 1 FADH2. There. Each has a pair. Look at this, look at the slide. Each has a pair of high energy electrons because this is NADH plus H. This is not a typo. Can someone explain that diamond on the bottom that I have right there? And this is why you got to read test questions with ruthless accuracy. And I talk about this in the Strategies for Success online tutorial on test taking. Read it with ruthless accuracy, and someone explain how this is correct and this is correct. Hint, read it with ruthless accuracy. It's not tricky. It's just, can you read with ruthless accuracy or not?
There's 2 hydrogens on the FAD.
They all have 2. So they all have a pair. Because this is NADH plus H. So each one is a pair here too. See, the trick here is I want to get you guys to be able to read with ruthless accuracy as a reflex. It's become so natural. It's as natural as breathing. Every exam you take after that is easy.
Is it because after the 3-carbon pyruvate, you have that extra NAD that's...
Boom. Exactly. The key here is from the original pyruvate. That means it includes the prep step. So these 4 pairs come from the Krebs cycle proper. But if you go back to the original pyruvate, you got this 5th pair. Why is it a pair? And just remember, well, it's because it's NADH plus H. But just remember, when you have oxygen, both pyruvates are doing this. That's the very last line on the slide. Everybody see that? You see it now. This is coming from acetyl-CoA. This, from the original pyruvate, includes the NADH plus H that comes from the prep step, the linking step.
And you would need to double this for the total net, right?
Yep.
Okay.
It would be 2 cycles per glucose. Does it help having it color-coded here on the whiteboard? 1, 2, 3, 4 from the Krebs cycle proper, 5 going from the original pyruvic acid. Those are not typos. I do this every semester because I want students to learn the importance of attention to detail. Could be the difference between you doing a good job or you killing somebody. Attention to detail. Yeah, see, you don't have to learn this from the textbook. This is the biochemistry. And we had to learn all the names of all these stinking enzymes too, as well as the formulas. But you got to know this. And this is essay number 4. Where does the Krebs cycle proper occur? What goes in? What comes out? Okay. And that's it for step number 2. Now notice on the whiteboard here, all the carbons are gone. Let's see, what color haven't I used yet? We had 6 carbons here, 3 carbons here. Out of that 3 carbon, here's 1, here's 2, here's 3. Now double it. 3 times 2 is 6. When you're done with the Krebs cycle, all the carbons of glucose are now carbon dioxide. And when trillions of cells are doing that, that's why when you exhale, it's pretty thick in carbon dioxide. And it's not only carbon dioxide, but there's a lot of that. So if there's no carbons left, how do we make more ATPs? This has to do with the high energy electrons on these electron carriers. Now I'm not going to be able to do it on all these, but that's where this is going. Okay. Every one of these high energy electrons of NADH plus H and even this FADH2, and pay attention because they drop it off someplace else. I can write that dashed line all the way over there to the right where it says number 3. And that's where I'm going to talk about the electron transport system. And that's this. This is the final step. And here we can now look at the inner mitochondrial membrane and recognize the fluid mosaic model of cell membranes, because there are protein boulders floating in this. Again, I just wrote 2 lines there. I'm not going to write out all these phospholipids. And then it's going to be this special one here. It's also a coenzyme here shuttling it in the middle. All right, hydrogen is just a proton and electron. So with these hydrogens, it's going to drop off the electron to the first boulder, and the proton's just going to go in the matrix. Because that's what H's are, right? Hydrogen is just protons and electrons. Well, this electron has got a lot of energy in it. And you can see that in this figure. But these boulders are in the inner mitochondrial membrane. So there are 3 main protein complexes. That's why I wrote 3 brown boulders. There's one little shuttling coenzyme in there as well, but don't worry about that. Now this high energy electron, once they accept the electron, this first one becomes reduced. Then it's going to pass on the electron to the next one. And it's going to pass on the electron to the next one. Okay. And eventually this guy's going to drop off this electron, but by the time this electron has gone through all those protein boulders, it's lost all the energy. It's getting lower and lower in energy every time it's passed, but it's getting passed from one to the next. But it's saying here that when you accept the electron, that first boulder becomes reduced. When it drops it off for the second one, it becomes oxidized, and the second one becomes reduced. When the second one drops it off to the third one, this becomes oxidized, this guy becomes reduced. And then eventually it drops it off here. I'm going to explain what happens to that electron once it's in the matrix. Everybody follow this? That's why it's called the electron transport system. These boulders are just passing the electron, transporting the electron from the next one to the next one to the next one, finally back into the matrix. Okay, so those high energy electron carriers, they got the H's, are going to drop off the electrons, go back to NAD and FAD. And then it can go, those NAD and FADs can go back to the first 2 steps and get high energy electrons with some more glucose. Okay, but again, they are first an electron acceptor, they become reduced, then they are an electron donor. They become oxidized. This is the redox reactions. Okay, and that's what happens to the final electron is actually it's going to happen twice. And it's going to join with a single oxygen atom. And that's why it's called oxidation, because oxygen is the final electron acceptor becoming water. Okay. So oxygen, an oxygen atom is the final electron acceptor. So it oxidizes everything else, which is where we get the term oxidation from. But it's 2 electrons along with 2 protons. Since there's a bunch of protons down here in the matrix, it's going to also add in 2 protons. That's 2 H's. So get this X out of here, that's confusing. 2 electrons, 2 protons, that's 2 H's, go on a hot oxygen, it becomes water. And in fact, when trillions of cells are doing this, after the course of a day, your cells make a measurable amount of water, like 250 milliliters of water from this, what I'm just describing. All right. But again, since there are no carbons here, this is oxidative phosphorylation. No substrates. Carbons are all done at step 2. Okay. Now, the electrons of NADH go to the first one. But if the electrons come from FADH2, they skip the first boulder and drop it off at the second one. And this is why, if you look at this figure, why you get more ATPs from NADH. Those pair of electrons go through all 3, and then you get 1, 2, 3 ATPs. FADH2 skips the first one, drops it off here, skips this one, and it goes down the line, and you only get 1, 2. This whole figure is, again, just like I drew it on the whiteboard, is going left to right. So if the high energy electrons start at the beginning, it gets 1, 2, 3 ATPs. But if they're dropped off here, it only gets 1, 2 ATPs. Everybody see that in the figure? On the bottom of page 10 is a scorecard. Don't worry too much about exactly where you get the counts. You should know the basics, and there's basics on the bottom of page 10. I've never really asked in lecture exam 1 really specific questions about that. And one of the reasons why I've never done that is because this 36 ATPs is what we would consider to be an ideal yield for glucose. 2, step 1, 2, step 2, 32, step 3, you got your 36. Well, no cell makes that much because there are costs here, ATP costs, that I'm not describing here. When a cell, a normal body cell is doing this, you might get between 28 and 30 because of the costs. We're just going to talk about ideal yields. Is that okay? Shall we try to keep things simple? You guys are probably thinking, this is not simple. Yes, this is simple. We're only going to talk about theoretical ideal yields. We're not going to talk about actual yields. But like your textbook, if you read your textbook, it likes to say 38 ATPs. And I'm going, well, okay, that's fantasy land, so let's just keep it at 36 and don't worry about it. Know that no cell even does that. It's actually more like 28 to 30, depending on the cell. But here's from your textbook showing you this. The electrons from NADH go through the whole box. 1, 2, 3. The electrons from FADH2 skip the first one. 1, 2. Does everybody see that? And then here it is. 2 electrons, 2 protons go on the oxygen, forming water. All right, now let's talk about essay question number 5 on that document. The chemiosmotic theory, which is question number 5, and I would write this down somewhere because it's not on the slide, but it's big picture comment. The chemiosmotic theory is a more accurate and precise explanation of the third step, ETS, electron transport system. The chemiosmotic theory is a more precise and accurate description of the electron transport system. Because I use this figure, and it's a gross oversimplification. Why is it an oversimplification? Because ATPs are not made at the site of the redox reactions. It is not made here, where the electrons are transferring from one boulder to the next. Those are the redox reactions. That's not where ATP is made. Instead, what is happening is that these protein boulders are actually referred to as proton pumps. Why? These brown circles here are proton pumps. As the electron is being passed from one boulder to the next, it's taking that proton from the matrix and pumping it into the intermembrane space from the matrix. So what I drawn on the whiteboard is very similar to what is in this figure, with the intermembrane space on top. So those protons is actually H+. And you get a bunch of H+'s up here. And let's say all these purple dots are H+'s. Why is there so much more H+'s up here? Because all those electrons, as it's being passed, these protein boulders are pumping against the concentration gradient from the matrix to the intermembrane space. That is what's actually happening at the site of the redox reactions, not making ATPs. Okay, that's what this bullet is saying. This creates both a pH and voltage gradient, what we call membrane potential. That's going to be in lecture exam 2. It takes a lot of energy to make it. It takes a lot of energy to maintain it. Because that H+ is the H of pH, isn't it? That's why there's a pH gradient. And the way the chemiosmotic theory works is all those hydrogen ions can't get back into the matrix except through these things called respiratory assemblies. And that's what I drew here. This is facilitated diffusion. Why? You got way higher concentration of hydrogen ion up on top, way lower in the matrix, and it's going through a protein channel. Boom. Okay. Then what happens? Again, it's small sequential steps. As the protons are flying through the respiratory assemblies, attached to the respiratory assembly, on the matrix side is this enzyme called ATP synthase. And as the protons are flying back through there, it's spinning ATP synthase like a pinwheel. You haven't had a county fair in a while. But sometimes kids get those pinwheels, and then when they're driving home in the back seat, they stick the pinwheels out the window, and it's spinning like mad from the wind. Okay. And on each rotation, it's doing 3 oxidative phosphorylations. Here in this animations, you can see ADP with the 2 phosphate groups. It's just putting the last phosphate group on ADP. And every cycle around, it's 3. This is one of the most energy efficient energy transformations in biological systems. Okay. An ATP synthase, because it is so efficient, so energy efficient, this has to do with laws of thermodynamics. With every energy transformation, some energy is lost. There's no such thing as 100% efficient energy transformation. This one's really high. So this has such high energy efficiency. This is why we're going to get 32 out of the 36 ATPs from this. Okay. You get 2 from the Krebs cycle, you get 2 from glycolysis, 32 come from this. So if you want to have ATP available for your cells to do work, you better have oxygen. Because remember, all the way in this middle board, you only get the 2 net ATP from glycolysis and anaerobic respiration, that's it. Because the NADHs that could have contributed to this get used up to make lactate. But if you have oxygen, you're going to get all the rest of it. Again, this is the summary. That's the kind of thing you want to have, the summary. You don't want to have all the very specifics. See, in your textbook is saying 34 instead of 32, and then they're saying there's 38 from the overall reaction. Well, that's ridiculous. Okay, there's various things there. Are there any questions on that? This is the framework. This is this 3 sequential steps going back to that first nutrient link, glucose. Let me know if any of these links don't work anymore either. Okay, so let's summarize before I go off, because the rest of this doesn't take that long, actually. Again, do not be intimidated. Take a step back and remember what all this is for. This is about how our cells use the 3 nutrient links to make ATP, the only energy that it directly uses. Here are the nutrient links. I'm starting with glucose, and I just finished the 3 sequential steps. Glycolysis, Krebs cycles, and the ETS. Then we fit the other two nutrient links in this framework. Because they do, they fit in this framework. What's the framework? You take a 6-carbon glucose and you split it in glycolysis. This is step number 1, happens in the cytoplasm. Can happen with or without oxygen, so I did have to cover anaerobic respiration. Step 2, if you have oxygen, is the Krebs cycle. It happens in the mitochondrial matrix. Now you know where that is. Remember that both pyruvates are going to do one or the other. It'll never split like that. I just don't have enough whiteboard. But all those purple guys with H's are going to be taking their high energy electrons to number 3. All the carbons are gone after step 2. That's the framework, starting from glucose. Any questions? Is this too much for you? Pharmacology is going to kick your ass if you think this is too much for you. So we're going to now fit the other 2 nutrient links in, and that would be lipids and proteins. We just did carbohydrates. Hey, this thing is not that intimidating. And this, I'm going to use this overall figure. We'll see this again. But in black, look at this carefully. In black is what we just covered. This is the framework based off of glucose. Blue are amino acids that go in this framework. Red, fatty acids and glycerol. We're just going to concentrate on that. That's where they fit into the framework. It doesn't do any good to talk about fats and proteins until I do this first. Fats, lipids. You do have to know the difference between lipolysis and beta oxidation. And I did mention this last week when I talked about ketones. Now, while glucose is the primary source of immediate energy to make ATP, fatty acids are the major primary source of stored energy. That's different. All right. I'm just going to draw this out here. Hopefully it'll make some sense. Hey, are you guys tired? I was here at 10:30 drawing on the whiteboard, so, and I'm the one jumping up and down, going all over the place, so don't give me that. All right, so remember triglyceride is a glycerol, and I'm just going to draw it like that, 3 carbons, and it's going to join up with 3 fatty acids, which are long chain hydrocarbons. So this is a triglyceride. When you split off with 3 hydrolysis reactions, let me change this a little bit here. You're going to split this up into glycerol plus 3 fatty acids. I'll amend that later in this semester. Everybody understand that? This is catabolism, correct? This arrow is lipolysis. It happens first. Now remember, these are long hydrocarbon chains, fatty acids. And so these are all, let's say this fatty acid is 16 carbons. Let's say, we'll just say this is a 16 carbon fatty acid. Now it's going to go through several reactions. That's why I'm drawing several arrows. And you're going to get 8 2-carbon molecules. Remember, we call this acetyl groups. 16, a long hydrocarbon chain is going to be 8 acetyl groups. This is called beta oxidation. And it's several reactions. That's why there's multiple. So here in this slide, it says lipolysis and beta oxidation, resulting in 2-carbon acetic acids or acetyl groups. And that's what I have there. One of the intermediate steps of beta oxidation is ketones. So I'm just going to apply that to that one arrow. And if you're doing a lot of beta oxidations, you're going to have elevated ketones. I will come back to that later on in the semester. But you got to know the sequence. If you're going to break down fats to make ATP, you got to take that triglyceride. You got to first go through lipolysis to get glycerol and 3 fatty acids. Each one of these is 16 carbons long, and that's just an example. There are other ones. It's going to go through a whole series of reactions we call beta oxidation to get you 8 acetyl groups. All you got to do with each of those acetyl groups is put a coenzyme A on it. And they go right into the Krebs cycle. If you put 8 Krebs cycles together, you're going to get enough NADH and FADH2s to get you 108 ATPs, because this is 8 Krebs cycles. Wow, I just did 1 fatty acid. There's 3 of them. And that's why triglycerides are a major primary source of stored energy. Okay? 108 with 8 acetyl groups. Well, that's got 3 of them. That's 324 ATPs for 1 triglyceride. Everybody got it? This is illustrating the central role of acetyl-CoA. Again, this green arrow is lipolysis. This green arrow should be several arrows. That's beta oxidation. Lipolysis first, then beta oxidation. Get you a whole ton of ATP. Okay, this, I was really bummed when the textbook dropped these figures from the 4th edition. I thought it was really good. What we did here first is we did this framework in the center. What I just described just now are the purple sides on the right side. Glycerol can be made into glucose, by the way. Can anybody tell me what this purple arrow would be called, since glycerol is not a carbohydrate?
Gluconeogenesis?
This purple arrow would be gluconeogenesis. Your liver can make glucose out of glycerol. Since it's 3 carbons, it puts 2 3 carbons together, it makes a 6-carbon glucose. All right. With proteins, you do have to recognize the amino acid link as NCC. And if you're going to put this into the framework we just built, you got to take off the nitrogen. That nitrogen is called an amino group. from which we get the term amino acid. When you take off that amino group, it's called oxidative deamination. Now all you have are carbons. And those carbons can be used in a variety of ways. But first you got to take off the nitrogen from the NCC. Now you just have CC, you got carbons. Okay? Well, what do you do with that amino group? Well, you could make other amino acids out of them. That's called transamination. Or you can take that amino group, put two of them together and form urea. Again, your liver is the one that's making urea. All your kidneys are doing is filtering the urea out of your blood. Your kidneys do not make urea. Okay? Your liver makes. Your liver is the primary organ of intermediary metabolism, which is why it is a top tier organ. But that's called transamination, when you're just moving that amino group around. Taking it off is deamination. Everybody recognize the difference? But those remaining CCs, the carbon chains from the amino acids, can be made into pyruvic acid, acetyl-CoA, and any of the, not any, but most of the intermediates of the Krebs cycles we call keto acids. Those are the intermediates of the Krebs cycle. And that's in those blue boxes. So if you have oxygen, you can make ATP from amino acids. And in this figures from the 4th edition, that would be over here in the blue arrows on the left. Pyruvate, acetyl-CoA, or the keto acids of the Krebs cycles. And boom, you can make ATP based off the initial framework of the 3 steps: glycolysis, Krebs, and ETS. And that's what you see here on this figure. Black is what we covered first, off glucose. Blue is where those amino acids, those are all abbreviations for the 20 amino acids, where they can go into that framework. Fatty acids become acetyl-CoAs. Glycerol can actually go into gluconeogenesis and go through glycolysis. Any questions? And I got it done in time. You need to see the whiteboards again. Now notice how this can be really intimidating. Don't be intimidated. Take a step back. Remember what this whole thing is about that we talked about today. It's about how our cells turn the nutrient links, the 3 nutrient links, we just finished them up, into ATP energy. We started with the 3 sequential steps with glucose. Then we fit the other 2 nutrient links in that framework. Right, there's the 3 nutrient links, 1, 2, and 3. We're only worried about 1, 2, and 3. We're not even worried that you actually can make ATP from nucleotides, but that's much more simpler and just something that we don't have to cover. Here's the framework. 1 is glycolysis. 2 is, oops.
Okay.
2 is the Krebs cycle. It has the prep step. But only after this, circled in blue, all the carbons are gone. So that's all the substrate phosphorylation you see on the screen. All the carbons are gone into the CO2s. Now all you got left are those purple H's. Where do the purple H's go? They go into the ETS. Now you have to know about the chemiosmotic theory because it is a more precise explanation of what is actually going on in the electron transport chain or system. Okay. Any questions? Monday Lab, I'll see you in about 40 minutes.